Hello students, just a quick video as I know some of you will be away for our Monday, May 23rd lesson due to leadership responsibilities. So I thought I'd put this together so that you can stay up to date. So today we're just looking at reviewing some key terms from Year 11 biology so that you can review what you have learnt last year and build upon that as well. Previously we've looked at how independent assortment and random fertilization can alter the variations in the genotype of offspring, and you looked at that through the process of meiosis 1 and meiosis 2. So let's have a quick jump in. Now this one can be a great review for you. This is a song. It's pretty fun, and I highly recommend playing it on, say, 1.5 speed to review what you have been doing, learning about meiosis in the last two lessons. So that's for you to do in your own time. And throughout the lesson as well, you'll see these little definitions of key terms. So I'm not going to be reading from them. They're there for you to remind you what they mean as we're going through. So looking at genes and alleles and the differences between them, well, going back and putting it all together, we know that the human body contains 100 trillion cells, and there's a nucleus inside each of our cells, except blood cells, of course, they're far too small. And each nucleus contains 46 chromosomes arranged in 23 pairs, and you looked at what that looks like as a karyotype. And we have one chromosome of every pair is from each parent, so a paternal and a maternal pair, singular to make the pair. And the chromosomes are filled with tightly coiled strands of DNA. Well, they are the tightly coiled strands of DNA. And genes are segments of DNA that contain instructions to make protein like building blocks of life. So that's how that whole sequence fits together. And they are segments of chromosomes, genes are, and they're found within the nucleus of every living cell. Now, the key difference here, I want you to understand what an allele is. So while the gene is a unit of DNA that transmits genetic information, an allele is an alternative form of a gene that is found at the same location on the same chromosome. Okay, so for example, different forms of a gene, such as two genes for hair color, one from your mom and one from your dad, and a single individual may only carry two, one on each of those homologous chromosomes. Okay. So looking at uniquely where you might see or how we depict those alleles. So what you are used to is this kind of image. So this is a couple of paired chromosomes, and they are homologous, meaning the same. Remember, we looked at that word, that term, homo meaning the same, hetero meaning different. So these are the same, as they have all the same genes in all the same places along their length. To make the structure of chromosomes a bit easier to understand, we draw them like this. Okay, we have, for example, and these are just one of many genes along our chromosomes. For example, a gene for making earwax, a gene for eye color, gene for making the protein that dissolves the webbing between your toes before you're born, very specific. So as you can see here, while both chromosomes have the same genes, they can have different forms of these genes. For example, the chromosome form of the gene for eye color will give you blue eyes, this one here, and this one has the chromosome form for brown eyes. So when we have these different forms for the same genes, we call these forms alleles. So this is the allele for eye color, and on each of the maternal and the paternal chromosome. So looking further into that, when we're talking about alleles, we have dominant and recessive alleles. So hopefully some of these terms are coming back to you from when you learned about them in Year 11 biology. Don't worry if you've forgotten most of it though. So if the two alleles for a gene are the same, the individual is homozygous for that gene. Homo is the same. If the two alleles are different, the individual is heterozygous for that gene. We'll look at this further in a second. And one allele can exert full control or dominate over another allele in a heterozygous condition, and this is the dominant allele. And a person with a dominant trait may be either homozygous or heterozygous for that trait. Well, what does that mean? If we have a look at this little image down here, homozygous, meaning the same, this little guy here, B and B, is the same, homozygous, and they're in uppercase, so we know that they are dominant alleles. And this one here, lowercase b, both the same, also homozygous, but when we write them in lowercase, we know that they are recessive alleles. Over here, we have two examples of heterozygous alleles. Okay, so we have an uppercase and a lowercase. Hetero meaning different, and a lowercase and an uppercase B, also heterozygous. Now try to get into the habit of writing the uppercase first, not second, as in this example here. It just makes it a lot easier if you're consistent with how you write your dominant and recessive alleles by putting that capital first in all instances. Okay. So you probably heard about Gregor Mendel in year 11, and he was a key, I don't know if you could call him a scientist as such, but perhaps he was. He studied natural sciences and mathematics in Austria. He actually tried to become a teacher, but he failed, and just ended up continuing with his famous pea plants. And his most famous experiments were done between 1857 and 1864, 65. And he grew so many pea plants and studied them. And he must have had, I think, over 10,000 pea plants. And because they both have male and female sex cells, they can fertilize themselves. But he also was able to individually crossbreed the pea plants by transferring pollen himself. And he did this using a tiny paintbrush. So he amassed a huge range of data, so of characteristics for each plant, including its height, its shape, the shape of its pods, the pea shape, whether they were wrinkled or smooth, and the color. And when the plants self-fertilized, these characteristics, he noted, remained constant or consistent in the offspring. So at the time, so back in the 1800s, it was believed that heredity worked by blending. So the characteristics of the male and the female, when they produced offspring, it was thought that they were just kind of diluted. So it wasn't until Mendel showed that when two varieties of purebred plants crossbred, the offspring resembled one or other of the parents, not a blend of the two. So from that, he himself found that the traits are dominant and would always be expressed in a first generation cross, while others which were recessive wouldn't appear in that generation. So he wasn't believed for a very long time. He made a lot of hypotheses, and it wasn't attributed to him until many, many, many years later. And thanks to his hard work and his diligence creating these experiments and collecting all this data, we now know through his studies that there are a couple of things called the law of segregation and the law of independent assortment. And he was just way ahead of his time. He actually, as I said before, his work was largely ignored for years, 30 to 40 years. And he actually hypothesized that parents contribute some particular particulate substance. Obviously, we didn't have the technology to see that back then, but he hypothesized that some particulate substance to the offspring, which then determined its heritable characteristics. We now know that these particles correspond to genes made of DNA. And he did this without knowledge of any of the molecules involved, and he inferred that those heritable particles are separated into sex cells or gametes, the eggs and sperms, and that offspring inherit one particle from each parent. And alongside that, he also arrived at the theory or the law of independent assortment, where he stated that each allele pair segregates independently during gamete formation, giving different traits an equal opportunity to occur together. So I put a little video here. It's nice and short. You can watch that yourself. And I'll go into more information about this right here, this Punnett square, in just a second, and you can have some practice with them as well. So underneath in the notes section of this slide, you'll find a link to this video here. So you can pause and go and watch that now. So some other terms that you really need to recall are things like genotype and phenotype. So there is another video here, very short, only a few minutes long. It's a great visual. So if you're a visual learner, stop the video, find the link in the notes section and go and watch it. But basically it's quite simple once you understand the difference. So the genotype is an organism's genetic information, and we show an organism's genotype by writing things like this. So showing if it's a heterozygous or homozygous dominant or homozygous recessive organism, then we show it using these letters. So this is its genotype. Its genotype is then expressed as an observable physical traits, and we call that phenotype. So a phenotype is what we actually observe. So you think of pH, phenotype, pH physical, what we physically observe. They're things like the color of a plant or the size or the shape of its petals or the color of your skin, or if you have freckles or not, or your height. Or okay, so genotype, the genetic makeup of an individual. We show it like this, and we can further define it as homozygous, heterozygous, homozygous dominant, or recessive. And phenotype is the actual expression of that genotype. Now, a little reminder that homozygous describes a condition in which both alleles for the gene are the same, and heterozygous, each allele for the characteristics exhibits a different expression. So we have a look at a Punnett square right here. We can see that the paternal or the male alleles are up here. We have the female over this side. So the male has an uppercase B and a lowercase b, and that means that because they're different, this is a heterozygous. And down for the paternal, we have again a uppercase b and a lowercase b. They are also heterozygous. Okay, so when we're working out if what expression or what genotype or phenotype is in this instance a flower might have, we can use these Punnett squares and we just cross-reference. We have the paternal B here, the maternal B here, and we know that because this is now homozygous dominant, meaning they're both the same and they're both the dominant are being expressed, we know that it will actually express that color, that purple color. Now, this white color down here is a recessive color, and that will only be expressed if both recessive alleles, the lowercase b and the lowercase b, are available. So the paternal, the maternal, we have a homozygous recessive flower right here. Remembering that an allele for a gene that overrides the effects of the recessive allele in the heterozygous condition is autosomal or sex-linked. And recessive alleles are found on a chromosome that can be masked by its dominant form in the heterozygous condition, and it can also be autosomal or sex-linked. So I'd like you to do a little bit of a practice on a Punnett square and see if hopefully all your memories come flooding back. So grab your paper and a pen and just pause it here while you get those. And I'd like you to construct and complete these Punnett squares. So there's 3. This is number 1, 2, and 3. I'd like you to identify if the parents are homozygous or heterozygous, and you can write that up here. So which parent is, what is this here? Is this homozygous or heterozygous? And along here as well. And then number 3, calculate the probability of having blue eyes. And then optional, if you would like to colour in the boxes to show the offspring's eye colour, you are welcome. So let's do one together. So if we have, let's say this is the paternal one, we have uppercase B, lowercase b, we would call this a heterozygous. I'll see if I can use this mouse. Oh, it's terrible using this mouse pad. I'm using my Mac, not my work computer. So this is heterozygous, and over here you can see it's the same B and B, and this is also heterozygous. So both parents are heterozygous. Now if we fill in the Punnett squares, we can see that this is B, uppercase B, and uppercase B, because you've come from this one and this one. So if we do this point here, we've got a uppercase B. Feel free to watch this on a faster speed as I navigate this pen with my mouse pad. Uppercase B, lowercase b over here. Again, we have our uppercase expression up here, the genotype, uppercase B and a lowercase b. And up here, the lowercase b with the lowercase b. All right. So you've constructed and completed the Punnett square. Identify if the parents are homozygous or heterozygous. You've done that. Calculate the probability of having blue eyes. As it says right up the top, Blue eyes are recessive. So recessive alleles are going to be shown with a lowercase b. And remember, a recessive trait will only show if both alleles are recessive, because in any other situation where you have the dominant allele, that will override or dominate over the recessive one. So for example, this one, where the phenotype will be brown, brown eyes are dominant. This one will also be brown because you have the dominant allele here, capital B. This one is also containing a dominant, which overrides the recessive, also brown. So over here, this is the only one that will be B. So this has a 1 in 4 chance in this particular situation, 1 in 4 chance of blue eyes being there, being formed. Okay, so pause this now, construct these in your books yourself, and we'll come back and have a look at the results. Okay, how did you go? So let's have a look at number 2. So our paternal is a lowercase, and the maternal is uppercase, lowercase. So for this one, it would be homozygous recessive. And for this one would be heterozygous. Okay, and if you were to fill all of these in, you would see that there would be a 2 in 4 chance once you fill all that in. So we're looking for, the question actually asks, color the boxes to show the offspring's eye color and calculate the probability of having blue eyes. So blue eyes are recessive, it's a recessive trait. So the only Punnett square area that you would have those 2 recessive alleles that would be able to then express the blue color would be down here, because this guy here is going to be in this box and also this box with the lowercase. And the dominant one always overshadows or dominates the recessive. So for this guy here, you have 2 chances out of 4. Or, as always in maths, try to reduce that down. It is a 1 in 2 chance, or a 50% chance. Okay. Now over on this one, number 3, again we have an uppercase and lowercase. This guy is heterozygous because he has 2 different ones. This one over here, both uppercases. So this one is homozygous, but also homozygous dominant. And the question is asking, calculate the probability of having blue eyes. Well, if we were to complete this Punnett square, we would see that the dominant, or the uppercase B, would be present in every single square. Therefore, it is impossible for any offspring from these 2 parents to actually have a child with blue eyes. Okay, so I'm just filling this in. Hopefully you're watching this on triple speed, because I know it's taking me a while. Thanks for your patience. So you can see in all of these, the dominant B, or the uppercase B, is present. So there is no opportunity for blue-eyed child to appear. Okay, so here's a couple of more review questions. Now, remembering to look up here at this part here. So we've got the following pea plants, we're going to call these. Pea plants being classified based on their genotype being expressed here. Just remember that round is uppercase R and dominant, and wrinkled is R and recessive, lowercase r. And the color of the peas, yellow, is an uppercase yellow, and it's dominant to green, which is lowercase y. So the question here asks you, are the following classified as heterozygous or homozygous? So just pause the video here and have a go. And come back to it. And if you have a look at the notes underneath, you'll be able to find the answers, but we'll go through the first one together. So we have an uppercase R. Is it heterozygous or homozygous? Well, they're both the same. So this is a homozygous, but it's also homozygous dominant because they're both the dominant alleles. This one here, we have an uppercase and a lowercase, so round and a wrinkled. This one has 2 different ones, so it's heterozygous. This one here is the 2 same, so they are homozygous. Homozygous recessive because they are the recessive allele. Although this one doesn't apply to this one, but this question does, down in number 2. If you have uppercase T, that is dominant, homozygous dominant, and then the uppercase T, lowercase t is heterozygous. You can also call this a hybrid, I guess. So down here now you can use this information up here at the top to help with question 2. What is the phenotype of the following peas? Remembering what phenotype means, that's the physical expression of the genotype. So what the species or the plant actually looks like. So if you have an uppercase Y and lowercase y, we know that uppercase Y is yellow and dominant, and even if there's a lowercase or a green there, it won't be expressed. So this plant will be yellow. This one here, uppercase R, lowercase r, is going to have round seeds. So the pea plant here will have round seeds. Lowercase y will be a green plant or green pea pod or green seeds. And this one, this is extra interesting because you have not only the color of the pea, but also the shape, or sorry, if the seed is round or wrinkled. So in this case, you can have a look at YY. It's going to be a yellow round pea plant because you have your dominant R's and your dominant Y here, overshadowing the recessive Y. Okay. So for this one, these next 2 questions, I'd like you to pause again and construct some Punnett squares in your books to determine the probability of these 2 producing wrinkled seeds and the probability of these 2 producing green seeds. So just pause the video now and have a go. Good luck. Okay, so I just drew this fabulous Punnett square here. Please do use a ruler in your books. It looks a lot neater and it's easier to review later on. So we have our parents here on either side, and the question is asking was the probability of these parents producing wrinkled seeds. If you see that wrinkled seeds are recessive, you know that you'd need a homozygous recessive expression for that phenotype to be shown. And there's only one opportunity for that to happen in this particular Punnett square. So you could say there is a 25% chance or a 1 in 4 chance as well. So depending on what the question is asking you, I would generally answer in a fraction, or if you do are better at percentages, do convert it to a fraction. And in question 4, it's asking what is the probability of the genotype YY, uppercase and lowercase, heterozygous, and a recessive or Y and Y, homozygous recessive producing green seeds. Seeds green are recessive, so we know that Y and Y needs to be expressed in order for that phenotype to be shown. And you can see in this particular Punnett square, we have 2 instances where both recessive alleles are being expressed. So there is a 50% chance of these parents or these parent plants producing green seeds, or a 1 in 2 chance, or 2 in 4, 1 in 2. So hopefully some of those expressions and those terminologies and definitions have come flooding back to you from year 11. A reminder that your book is a good source of information, plus any additional links that I put in throughout this presentation in the notes portion. Please read page 218, 221, answer questions 1 to 3. For those in class today, we'll be doing a Kahoot, but we can possibly hold off till you get back or take a vote. All right, have a great evening.